Okay, let's get started. Hi, everyone. My name is Derek. I'm a business development manager at Immunodeficiency. It's a pleasure to welcome everyone today to today's webinar. We're excited to have you here with us today for today's talk titled Development and Application of Immunodeficient Mice. Before we get started, just want to quickly introduce our company, Genobiotexis, for those who may not be familiar with us. So at Genobiotexis, we're committed to advancing biomedical research by providing innovative and high quality animal models that can help accelerate the development of next generation therapeutics. Our goal at Genobiotexis is to help bridge the gap between basic research and clinical application. And we're excited to share today's insights that can help you achieve those. In today's session, we're going to cover the following topics. First, we'll introduce the development history of immunodeficient mice. Next, we'll cover the features and applications of each generation of these immunodeficient mice. And finally, we'll present several case studies using our models at Genobiotexis, specifically these immunodeficient mouse models. Also at the end, we're going to have a Q&A session. So if you have any questions throughout the webinar, please feel free to drop your questions in the chat, and then we'll be sure to answer them at the end. So I'm thrilled today to introduce our expert speaker, Dr. Haili Zhang. Dr. Zhang earned her Master's of Science in Biochemistry and Microbiology from Shenyang Agricultural University in China in 2003. And then she later got her PhD from the Institute of Genetics and Developmental Biology at the Chinese Academy of Sciences in 2007. She has over 15 years of experience in genetics, genomics, and infectious disease research. Dr. Zhang has worked in both academic and industry settings. Her expertise includes developing high throughput in vitro assays and platforms for drug target identification and efficacy evaluation, as well as using immunodeficient mouse models for in vivo drug targeting validation. Currently, Dr. Zhang serves as a business development manager here at Genobiotexis. And without further ado, let's get started. Dr. Zhang, the floor is yours.
Okay, thank you for the Derek's introduction, and thank you again for joining us. Again, my name is Haili Zhang, a business development manager at Genobiotexis, located in the Houston area, Texas. So we are also known as Shanghai Model Organism Center, SMOC for short. So we know immunodeficient mice are invaluable tools in research, especially in fields such as immunology, cancer, gene therapy, and infection diseases. Today, we will explore essential information about various immunodeficient mice and their applications, which will help you to choose the most suitable immunodeficient mice for your research. So this is the outline of this presentation. I will begin with a brief introduction to the development history of immunodeficient mice. Then we will go over the features and the applications of various immunodeficient mice. Following that, I will provide specific information about NMG and its derived immunodeficient mice, which were developed by our company. Finally, I will give you a brief overview of the company, the products we offer, and the service we provide. The history of immunodeficient mice, in fact, is the story of how research have optimized these animal models to study human diseases and the development therapeutics. While humans and the mice are two distinct species, they share some similarities, particularly in the functioning of their immune systems. This similarity had made mice invaluable tools in biomedical research. Here list some similarities between these two species. The overall gene count is almost identical. It's around 20 to 25,000 genes. And also human and mice share about 85% of their genes. And the conservation of homologous genes is quite high. For instance, many genes involved in immune response, nervous system function, and metabolism are highly conserved. On the right, an image of hypoidism, a rare condition in humans caused by a mutation in the KIT gene. The image demonstrates how a mutation in the KIT gene can produce a similar phenotype in mice. This animal model can be used for a tool for study this human disease. However, we know that there are some significant differences between human and mice. First, the mice immune system is different from the human immune system, especially in terms of immune cell populations, receptors, and the response to infections. Additionally, some human diseases, such as neurodegenerative disease and certain cancers, are highly complex and influenced by many environmental and genetic factors, and also some chronic diseases in humans, such as cardiovascular disease or autoimmune disorders, takes years to develop in humans, but mice typically have much shorter lifespans. So most models may not capture all aspects of certain diseases. One strategy to address this limitation and optimize animal models is the development of humanized animal models, allowing for more accurate representation of human disease processes and responses to treatment. So what is a humanization? Basically, there are two approaches for humanization. One is a gene humanization and another tissue and cellular humanization. So for gene humanization is an approach of using gene editing tools such as CRISPR-Cas9 or ESL-based technology to introduce one or more human genes into the mouse genome. This technology allows research to create mouse models that better replicate human genetic traits. These models can be used for simulating human diseases, tumor immunity research, and preclinical drug evaluation. So for the tissue cellular humanization, we transplant human cells or tissues in mice, which can develop normally and perform corresponding functions. So there are two key factors for a successful tissue or cell transplantation. First, we have to use immunodeficient mice to reduce the immune rejection. Sometimes we have to develop a humanized environment and to support human tissue and cell colonization or function. So we will talk about some of these models later. So let's review the history of immunodeficient mice. The discovery of immunodeficient mice has progressed through 4 major stages, resulting in 4 generational mice with increasing levels of immunodeficiency. So first generation is new mice developed in 1962, which carries mutation in FOXN1 gene. These mice lacks of T cell. Second generation is SCID mice identified in 1983. So these mice carry a mutation in PRKDC gene. These mice lack a T cell, B cell, but maintain the functionality of innate immune system. The third generation is not SCID mice. No SCID mice discovered in 1995, which carry a SCID mutation on their not mice background. These mice lack a T cell, B cell, and showed reduced macrophage, NK cell, and complement activity. So the fourth generation is a not SCID IL-2 receptor gamma knockout mice developed in 2002. This mice carries a SCID mutation and a IL-2 receptor gamma knockout under the not mouse background. As a result, these mice lack T cell, B cell, and NK cells, and also exhibit reduced level of macrophage, dendritic T cell, and the complement activity. So in 2017, Smoke independently developed a not SCID IL-2 receptor gamma knockout mouse model named NMG. Since then, we have continued to develop NMG-derived models to enhance the performance of human immune system reconstitution. These models are being used for more accurate evaluation of therapeutic drug efficacy and providing a valuable platform for research in immunology and drug development. So we will talk about these models in detail shortly. So let's go review some detail on each generation immunodeficient mice and their application. So nude mice carry a mutation in the FOXN1 gene. FOXN1 is a transcription factor that controls the expression of genes involved in thymus and hair follicle development. So the phenotype of nude mice shows hairless and thymus, growth delay, and decreased reproductive capacity. As a mouse model, hair presence or absence does not affect tumor engraftment rate, but no thymus affects the human and selection of T cell progenitors. So here shows two commonly used nude mice models. Nude mice model can be used for xenograft transplantation, cancer research, and immunology research. These are two commonly used nude mice models. One is new mice. It's a black 6 background, carry a homozygous mutation in the FOXN1 gene. Another is Bob C nude mice, Bob C background carrying mutations in FOXN1 gene. So this model was developed by our company. So here list the comparison between new mice and Bob C mice. So I would like to highlight this point. Bob C nude mice showed high genetic homogeneity, which lead to greater consistency and repeatability in test results. So for the xenograft model development, Bob C nude mice require smaller tumor cell inoculation, approximately 1 million compared to 10 million in new mice. Additionally, Bob C mice tend to show faster tumor formation with tumors developing approximately 1 to 2 weeks earlier than new mice. So this makes biopsy mice, nude mice, a more efficient choices for studies requiring quicker tumor progression. So in the following example of application of new mice, we refer to our company-developed biopsy new mice. So for the new mice, can be used for CDX-based drug efficacy evaluation. So on the left, tumor growth and metastasis in new mice are shown here. Here we use a B16 LUC cell line. This is a black 6 background melanoma-derived cell line that expresses luciferase, allowing for real-time tracking of tumor progression and metastasis. So from the figure here, you can see the tumor growth and metastasis after days of post-inoculation cells in the new mice. So on the right here, we use this model to evaluate drug efficacy in controlling tumor growth. The results show that the combination of compound D1 and D2 significantly inhibit tumor growth. So the controls group is this blue dots, and the green dots represented the combination compound D1, D2 treatment. So skin mice was discovered in 1983. It carries a mutation in PRKDC gene. So PRKDC is a DNA-dependent protein kinase catalytic subunit. This gene plays a critical role in DNA double-strand break repair. This gene also involved in VDG recombination process. So the mutation leads to lacking of T and B cell, but mice maintain the functionality of innate immune system and a normal development and differentiation environment for immune cells. So skin mice can be used for xenotransplantation, cancer research, immunology research. But there are two limitations of this model. First, since these mice have defects in DNA double-strand break repair, they are sensitive to irradiation. Second, some mice may produce a small amount of T and B cell and immunoglobulins, leading to immune weakness. So to overcome these limitations and improve the utilization of this model, scientists got ideas by understanding the mechanism of VDG recombination. So here on the right, the diagram shows the VDG recombination process, which involved several important proteins for DNA recombination and the generation of diverse immune receptors. Among of these proteins, the first pair is RAG1 and RAG2 protein. So RAG1 and RAG2 are responsible for the cleavage of DNA at specific recombination signal sequences that flag the VDG gene segments. And after the RAG complex cuts the DNA, the end of the DNA segment are joined by PRKDC and other proteins to form the functional VDG arrangement. So the mutation in RAG complex can disrupt VDG recombination and cause defects in T and B cell production. Then the scientists attempt to develop RAG knockout mice, which can disrupt the VDG rearrangement, but can overcome the shortcoming of the SCID mice, which is sensitive to irradiation and immune weakness. So the first RAG knockout mice was discovered in 1992, which carried RAG genes knockout alleles. So both RAG1 and RAG2 are key proteins involved in VDG recombination. So RAG1 has a nuclease activity and is essential for making the DNA double-strand breaks. And RAG2 regulate RAG1 function and is also crucial for the proper coordination of the recombination process. Here on the upper right is a flow cytometric data. So in this study, we detect B cell and T cell in the peripheral blood, a wild type RAG knockout and not SCID mice. The results indicated that both RAG1 knockout and RAG2 knockout mice had a significantly lower level of T cell and B cell compared to wild type mice. Additionally, this on the lower right, we evaluated the engraftment tumor growth in these models. The data showed that the tumor growth was significantly rapid in both RAG1 knockout and RAG2 knockout mice. In RAG knockout mice, there were no significant differences in tumor morphology or growth rate following tumor transplantation. So this model can be used for xenotransplantation, cancer research, hematopoiesis, and toxicology research. We have talked about SCID mice, RAG knockout mice. The research shows that the transplantation rate of human tumors in both of these mice is significantly higher than nude mice, but it hasn't met expectations. So in addition, studies also show that human PBMC, HSC, and embryonic tissues can engraft in those models, but are non-functional. To further explore strategies for optimize SCID mice model for human immune reconstitution, this publication data provide insights into potential approaches. So in this study, researchers use a compound to deplete the macrophage in SCID mice, and resulting in a significant increase in the engraftment of human PBLS compared to the control group. So this indicated that the retained innate immune activity is the main reason for preventing human immune reconstitution. So here, to increase the engraftment rate of human cells and tissues, the research tried various background mice for SCID mutation. So including black 6G, BOT-C, C3H, until they found a not background mice and generated not SCID mice. So Node mice are one of the most widely used animal models for studying human type 1 diabetes. So the mice display several immune system deficiency, including free complement C5, and also shows macrophage and other antigen-presenting cells functional defects. And another significant feature of this mice is that it shows high affinity of SIRPR for human CD47. So SIRPR is signal regulatory protein alpha. Is an inhibitory receptors primarily expressed on various immune cells, such as macrophages, dendritic cells, neutrophils, and others, which can inhibit phagocytosis by binding to CD47. So node background mice could show a high rate of human cell and tissue engraftment. So the first node skin mice was reported in 1995. The mouse backgrounds not carrying a PRKDC mutation, SCID mutation. So here list some feature of this mice. So this mice lack B cell and T cell and reduced functional AP antigen present cells and also macrophage and NK cells. On the right is a flow cytometric data from our company developed node SCID mice. So this data shows that node SCID mice here lacks of T cell, B cell, but still shows NK cells, which is comparable to biopsy, this Y-type mice. So this model can be used for PDX, based drug efficacy evaluation. The figure shows the drug efficacy evaluation use human liver tumor implanted into the node SCID mice. The drug NGS significantly inhibit the tumor growth and also increase the mice survival rate. This model can also be used for CDX models and the related pharmacology and efficacy study, hematological and stem cell research, HIV and hepatitis research. So the figure below showed you the tumor growth when inoculate cancer cells into the node SCID mice. The tumor rapid growth, and also here show you the mice survival rate increased after the drug treatment. So on the right, the table shows you the most popular cell lines for CDX models developed by SMOC. You can request those cell lines information through our website or any of our BD colleagues. So the major limitation of the node SCID mice is a residual innate immune activity. So on the right side, flow cytometric data shows that node SCID mice lack T and B cell, but still have comparable amount of NK cell monocytes and macrophage compared to the biopsy Y-type. So also the function of the NK cell is only partially compromised in this mouse line, which is also confirmed by this published paper. In addition, the mouse have a shorter lifespan. Average lifespan is 8.5 months under SPF environment. So how to optimize not-skin mice for better use is their challenge. Until the discoveries mechanism of the X-linked skid in mice, this in human. The significant feature of this X-linked severe combined immunodeficiency in human is a patient lacks of T cell and NK cells. So in the early 1990s, research sequenced the IL-2 receptor gamma gene and confirmed that mutation in this gene was responsible for the X-skid human disease. So gamma chain, interleukin-2 receptor, the subunit gamma chain, is a shared subunit in the receptor complex for multiple interleukins. For example, IL-2, IL-4, IL-7. So it is crucial for the signaling of these cytokines. So knockout gamma chain can significantly influence our immune system functions and responses. So the IL-2 receptor gamma knockout was first reported in 1995. The T cell and the B cell development and function are severely impaired, and also NK cell development is completely blocked in this knockout mice. This diagram here highlights the evolution of highly immunodeficient not-skid IL-2 receptor gamma knockout mice. So starting from the original skid mice in the CB17 background and crossing with a not-skid, then we obtain the not-skid mice. Then we cross the not-skid with IL-2 receptor gamma knockout mice. Then we obtain the not-skid IL-2 receptor gamma knockout mice. So this model is a crucial tool for human immune system research and tissue transplant studies. So the first not-skid IL-2 receptor gamma was reported as an OG in Japan in 2002, then followed by NSG, developed by Jackson Lab in 2005. So we also independently developed the not-skid IL-2 receptor gamma mice named NMG in 2017. So in this third section, I will mainly focus on our company developed NMG and its derived strain. So smoke NMG. The background mice is a NOD, and the mice carry two mutations. One is a PRKDC mutation, and also carry a completely non-allele of IL-2 receptor gamma chain. So the main feature of this model includes low immune rejection to human tissues and cells, and also the tumor genicity is high. The model significantly improves survival rate for the human cells and tissue transplants. So on the right side, the figure shows a comparison of tumor growth in biopsy, not-skid, and NMG mice, following the injection of same amount to human cancer cells. The results indicated that tumor growth in NMG mice is significantly faster than in both the wild type and the not-skin mice. And this MNSG is also another name for NMG mice. So it's same mice, different names. So additionally, for certain cell lines, such as GLMT1, a human breast cancer cell line, the tumorigenicity of this cell line is very low in other mouse models, but NMG mice can support the tumor growth. Here, the flow cytometric data shows that NMG mice lack of T cell, B cell, and NK cell in spleen and peripheral blood. And there is no any detectable immunoglobuline antibodies in serum. So the NMG mice can be used for PDX model-based drug efficacy evaluation. Here is an example. So in this study, the client successfully transplanted a human oral tumor to the NMG mice. We can see the tumor rapid growth after inoculation in the mice in the control group, in the PBS group. And in the treatment group, especially under treatment combination of several immunotherapy approaches, the tumor growth was significantly inhibited here and shrinked, and the expression of interferon gamma was increased under the treatment group. So the NMG mouse can be used for CDX model based drug efficacy study. So in this study, we injected 5 million this lung cancer cell into NMG mice by day 7 post-inoculation. Tumor was visibly detected in all groups, and the tumor growth progressed rapidly in control group. However, in the CAR-T treatment group, tumor growth was inhibited, and the tumor was eventually eliminated. So besides the CDX and PDX model for drug efficacy evaluation, NMG mice can be used for human immune system reconstitution. So there are 2 ways to do this. One using human PBMC, another use human HSC. So here is a simplified diagram, shows a procedure for the development human PBMC reconstitution mice model. So human PBMC are isolated from whole blood, then the purified human PBMC enriched with T, B, and monocyte and NK cells are injected into the adult NMG mice. The mice will reconstitute human immune system within 1 to 2 weeks. And we can use a flow cytometry to value the presence of human immune cells in the mouse blood, spleen, and other tissues. So once successfully reconstitution is achieved, the humanized mice can be used for various studies. Here list some, for example, testing immune response to infections, cancer therapies, or evaluating novel immunotherapeutic drugs, and also others. Here is a flow cytometric data show you the human lymphocyte subpopulation in humanized PBMC reconstituted NMG mice. So we can see the mice produce various human immune cells, CD4, CD8, NK cells. So then we use this immune system reconstituted NMG mice for CDX-based drug efficacy evaluation. So in this study, the human PBMC were engrafted 11 days before the treatment, and the tumor cell was inoculated 7 days before the treatment, and then the drug was administered at day 0. We can see from the figure below, the tumor growth was rapidly in the control group. However, it was inhibited by the treatment. So the immune system reconstituted NMG mice can also be used for PDX-based drug efficacy evaluation. So in this study, the patient tumor was transplanted into NMG mice. And briefly, we can see the tumor rapid growth under control group, and also but significantly slow under treatment, and also this improved the survival rate of the mice. So as mentioned, we can also use human HSC for human immune system reconstitution in NMG mice. Here's a simplified diagram, shows the procedure for the development of human HSC reconstitution mice model. First, human hematopoietic stem cell are typically isolated from umbilical blood or bone marrow. These cells are enriched for isolation CD34 positive cells. Secondly, prior to the injection human HSC, the recipient mice are conditioned using radiation or chemotherapy to suppress their endogenous hematopoietic system. Then the isolated and purified human HSC are injected into the conditioned mice. So typically by tailwind injection. So the mice will reconstitute human immune system within 12 to 16 weeks. The application of this immune reconstituted mice is similar to the PBMC reconstituted mice model. Here also, here's a flow cytometric data. Also show you the lymphocyte subpopulation in human HSC NMG mice. We can see the T cell, CD4, CD8, and other immune cells expressed in these mice. Similarly, human HSC NMG mice can also used for CDX and PDX-based drug efficacy evaluation. However, human immune system reconstitution in NMG mice can cause GVHD. It's a graft-versus-host disease. The basic reason for this disease is a transplant immune cells recognize the recipient tissues as foreign and attack them. So in mice, this disease can cause a poor skin condition, significant weight loss, and even death. So there show you some key factors that could affect the severity and outcome of this disease. For example, number of transplanted cells, whether irradiation was performed prior to transplantation. So to reduce the occurrence of the disease, we developed the B2M-KO-NMG mice. So beta-2 microglobulin is a small protein that is part of the structure of MHC class 1 molecular. It's required for stable expression of MHC class 1 molecular on the cell surface. So in B2M KO mice, the deletion of the B2M genes prevents the expression of functional MH class 1 molecular. This causes significant impairments in CD8 T cell response and the adaptive immune system. So on the right side of the flow cytometry metric data show that B2M KO mice lack of MHC1 molecular on the immune cell surface. This is another NMG-derived model. So to promote the development of NK cells and the proliferation T cells during immune reconstitution, so we developed this NVG human IL-15 mouse model. So in this mice, we replace the mice IL-15 exon 4 to 8 with human exon 4 to 8, and the mice can produce human IL-15 protein. So in this study, human pancreatic cells were engrafted into a VG mice 2 weeks prior to the CAR-T treatment. The figure on the left show you a significant increase in NK cell numbers in the NVG mice after 1 week of treatment compared to the control NMG mice. So this result is consistent with the observed efficacy of CAR-T treatment in NVG mice. Here, this table presents various NMG-derived mouse lines developed by Smoke. So this include human gene knocking models such as L2, L3, which enhance immune cell generation, proliferation, and differentiation following immune reconstitution. We also developed some gene knockout models such as MHC2 knockout, MHC2 B2M double knockout, which help reduce GVHD after immune reconstitution. So this table, I use this table to visualize the immunodeficiency difference among various models, which may help you to memorize the features of each model. So we know Bobsenian mice lack of T cell, skin mice lack of B and T cell, and not skin lack of T and B cell, and impaired innate immune system, NMG lack of T, B, and NK cells. Okay, the last section, I will briefly review some information about Genobiotechs. So our vision is to provide global researchers with professional, comprehensive, and integrated animal model services to help you to uncovering the mysteries of life. So this is our state-of-art housing facility. These are show you some our RD platforms, and this is a phenotypic analysis platforms. So we provide our clients one-stop shop services. So this include we can provide a customer customized model development services. We provide the off-the-shelf mouse lines, and also we provide breeding services and phenotypic analysis. So for the customized model development, we support knockout, condition knockout, knock-in, point mutation, targeted overexpression, and random transgenic model development. So for the off-the-shelf mouse lines, we have humanized mouse models, immunodeficient mouse model, fluorescent reporters, and disease models. And we can do the rapid colony development, drug screening, efficacy evaluation, and I have talked about a lot the CDX, PDX models. So for the in vivo PD evaluation, we target various disease areas, including cancer, metabolism disease, autoimmune disease, and nerve system disease. And also we provide various models for this PD, in vivo PD evaluation. And at the bottom, I just list some specific assays we can support those evaluation or development. For example, in vivo pharmacological assays, pathological and toxicological assays, in vitro pharmacology, all kind of these assays. So we serve global clients from academia and the industries. I think this all I want to share with you. Thank you for your attention. I'm happy to take any questions you may have.
Thank you, Hailey, for the great talk. So I don't see any questions currently in the chat right now. If anyone has any questions, feel free to put them in there and we can answer them. As well as I did notice some people got here a little bit late. Don't worry, we will be putting today's webinar on our YouTube channel, so be sure to check that out in a couple of days when it's uploaded. But if you have any questions, feel free to ask them. Okay, so we do have a question. What ethical concerns arise from using immunodeficient mice in research, and how can they be addressed?
What are other concerns? Is that the question?
What are some ethical concerns that arise from using immunodeficient mice in research, and how can they be addressed?
That's a good question.
I think I would say that in terms of the ethical concerns of using animals in research, all of our animals are held to ALAC and OLAB stringencies, so they're all maintained in the most sterile environments, and they're well maintained and cared for, to the extent of which we can provide the important research and studies that hopefully can help patients in the future. Okay, I don't see any other questions so far. If anyone who is here today has any other questions, please feel free to reach out to us at the email that you see on your screen, service.us@modelorg.com. We'll put you in touch with one of our scientists like Dr. Zhang or myself, and we'll be sure to follow up with you. With that, thank you for coming to today's webinar, and have a great rest of the week.
Thank you.